A warm welcome, everyone. Sorry to keep you waiting, but with schools being quite so busy as they are obviously this week, it's quite difficult for people to come on any earlier than this, but well done those of you that have made it. And this is our third day to celebrate British Science Week 2021. And all the talks will be recorded, especially because of the problems that schools have got at the moment with being quite so busy with starting back. It makes sense for all the talks to be recorded. So all of you that have obviously registered to attend, you will receive the link next week for the recording so that they can be used whenever it actually suits. Alongside me on screen is another member of the committee, Jolanta, who was very busy on Friday giving one of the talks of the trio that launched our event. And I'll pass you over to her in a moment. There is another member of the committee in amongst the audience. Jeff Butt, our treasurer, is there as well. Because of the recording, all of you will be permanently muted and your videos off. But please feel free, especially after the talk, obviously, to feed in some questions into the chat. Anyway, I'll pass you over to Jolanta.
Thank you very much. My name is Jolanta. I am the RSB ambassador for East Midlands, and today it's really great to see you all here today, and hi to everybody who will be watching this in the recording. Today, it's my great pleasure to introduce my very dear friend, Dr. Sam Horrell. He's a crystallographer and a beamline scientist. He's a leader of the crystallographer community on Twitter. He is Dungeons and Dragons Dungeon Master Extraordinaire, and today he'll tell us more about his career and research in biophysics. Sam, over to you. I will stop sharing my screen now so you can share the slides.
Okay, thank you for the introduction. So, like Jolanta said, my name is Sam, and I am an X-ray crystallographer, which probably doesn't mean a lot to most of you because it's not. I can't imagine it's something you're going to be taught in school. So before I go into the details of what X-ray crystallography is, I was told to introduce myself first. So my name is Sam Horrell. I identify as male. Pronouns are he, him, and I'm 31 years old. I was born in Bedford in the UK, and I have a Bachelor's of Science in Medical Biochemistry and a PhD in Biological Physics. I'm a first generation academic, which means first person of my family to go to university and enter academia. And my job title is Beamline Scientist. And if you are also on Twitter, you can find me at Dr. Horrell. So how did I get to have the random title of Beamline Scientist? Well, I started off in Sharnbrook Upper School, where I got fairly average grades, if I'm honest. Did pretty well in science, average in maths, and sort of not so good in chemistry at my A-levels. Maybe I should have just taken history instead. But after my A-levels, I actually applied for medicine and didn't get in. And instead, I was offered the opportunity to take a medical biochemistry degree at the University of Leicester, which I took and got a 2-1, which is 60% and above total in the exams. After my bachelor's degree, I went straight to a PhD. I managed to skip doing a master's, and I went to the University of Liverpool, where I started my PhD, and I then, as part of my PhD, it involved a 2.5-year placement down at Diamond, where I actually currently work now. So I spent 2.5 years in Oxford before moving back to Liverpool to spend 6 months writing up my thesis, which I think at the end of it was about 72,000 words and 330 pages, so a fairly big undertaking. After graduating, first I went traveling. I bummed around Europe for about 2 months, and then came back and started my first postdoc at the University of Essex. I was there for 1.5 years before moving all the way over to Hamburg, where I did another 3-year postdoc job. Then after Hamburg, I actually moved back to Diamond and started my 3rd postdoc, which lasted about 1 year, and then I applied for the Beamline Scientist permanent position at Diamond, which I currently occupy. So, as well as working all over the UK, science has also taken me to quite a few other countries. So I've been to conferences up and down the UK, and also I've been to Sweden and Austria, Croatia, Germany, France, over to the States, to California and New York and Maine. And we've actually got some experiments planned in Japan if we can ever actually get back to flying across the world again. So science can take you places, not just in your career, but across the world. So that's enough about me. You're here to learn about some science. So when I generally say I work with X-rays, what people generally think is this sort of thing, your typical medical X-ray. And this is obviously where X-rays started. This picture is actually from, I think it's 1885, and it was taken by Wilhelm Röntgen, who discovered X-rays. And what's the first thing you do with an unknown type of radiation you've discovered? Obviously, you put your hand in it. Well, you don't put your hand in it. You put one of your. I've heard tale of it's either his lab assistant or his wife, whose hand he actually first X-rayed. The other thing people also think when I say crystallography is some sort of magical crystal healing. And I can confirm that as a crystallographer, I have never artfully arranged crystals on a woman's head to try and relieve their headaches or anything like that. So what I actually do, and I'm going to go through each of these 6 steps in a bit more detail, is I grow bacteria to get at specific proteins. I then crystallize those proteins, which we bring to particle accelerator. This here is Diamond, where I'm working. We then fire X-rays at them, which produces this diffraction pattern. And from this diffraction pattern, we can calculate our protein structures. So let's start at the beginning with growing bacteria. So typically in crystallography, we will have a protein that we're interested in. So what we do is we take a circle of DNA, which is called a plasmid, and we generate our gene of interest. And so this is just a little section of DNA. We'll cut open our plasmid, so it produces these sticky ends. We'll cut our gene in the same way, and then we'll insert it into our plasmid. So within our plasmid, we also have this little region which conveys antibiotic resistance, and this other section which gives an expression activator. So we implant our plasmid into our E. coli, which is actually sounds really cool when you say, oh yeah, I spent today genetically modifying something, but all you really do is you mix them together and leave them on ice and then heat shock them a little bit, and it kind of shocks the E. coli and it opens up its pores and the plasmid goes in. So it's not nearly as exciting as it sounds. We then grow our E. coli in some growth media with an antibiotic present. So what this does is this kills off any other bacteria that might be in there, and means that we're just growing the E. coli that we've transformed, that we're interested in. Once this has grown to a high enough density, we add our expression activator chemical, which pretty much kicks off the transcription and translation of DNA into proteins. So after sort of 10 to 16 hours, we end up with our growth media full of bacteria that have been expressing the protein we're interested in. So we then centrifuge this solution, which makes these horrible smelling pellets, which I showed earlier. Actually, this is what this looks like after centrifugation. So then you have to scrape out the E. coli pellet, and then we subject the cells to either sort of high pressure, certain chemicals, or sound waves. And this just bursts open the E. coli and releases all the proteins that we're looking for. But obviously, an E. coli cell is made up of lots and lots of proteins. So we then have to go to the second step, which is purifying our protein. So a very common way of purifying our proteins is by something called affinity purification. So we have here a column which is full of little beads which have nickel ions attached to them. I should also say we're going to visualize our proteins using this gel here. So what we'll do is we'll add our protein solution into these wells, we'll apply a voltage, and it will display all the proteins that are present based by size. So the smaller the protein, the quicker it travels through the gel. So then we have this ladder as a means of judging the size. So what we're going to do is we take our protein, we run it through our column, and what's going to happen is our protein, which we've genetically modified to have this little tag on, is going to bind tightly to this nickel resin, and all the rest of the proteins that are in there are hopefully going to flow just straight through and not bind at all. But in reality, other proteins within the cell, or within the E. coli, will have amino acids that will also interact with these nickel ions, so there will be some contaminants in there. So we give it a wash, another wash, and we start getting rid of our contaminants. And then eventually, once we're happy with the amount of washing we've done, we'll add a chemical called imidazole. So you can see this chemical structure of this is very similar to this histidine tag. So what this does is it outcompetes our histidine and just pushes off our protein. So as we start adding imidazole, I've colored this band here green to represent the protein we're interested in. And then as you increase your imidazole concentration, you get even more of your protein coming off until most of it has come off. And then even with the higher washes, you'll just be getting a little bit. The next step is actually to reuse this column again and use it to our advantage that contaminants also bind to nickel. So we're going to cut off this nickel tag, and then we're just going to run it back through the column. And in theory, our protein from these three elutions, which we've pulled together, cleaved, and put back through the column, we should see our protein coming through and hopefully fewer contaminants. But it's very rarely the case that this purification alone will be enough to get pure protein. So we go to a second purification step. So this column doesn't have any nickel or anything in it like that. Instead, this kind of acts as like a molecular sieve. So it's full of these little beads which have a sort of a certain sized hole in them. And then when we run our protein solution through, what we'll see is the very heavy stuff will come out really quickly. This might sound kind of counterintuitive, but what happens is the really heavy stuff goes straight through the column and it doesn't end up falling into any of these holes, so it doesn't get slowed down, so it just goes straight through. And then at some point, after we've ran enough of our solution down, these columns are typically 120 milliliters in volume, you'll start seeing your protein that you're interested in coming off. And then hopefully right at the end, you'll see the smaller contaminants finally coming off. And then you'll be left with your nice pure protein. So once we've got our nice pure protein, we then go to the next stage, which is crystallization. So how do you crystallize protein? So the typical method is by this vapor diffusion method. So what we do is we take our protein solution and a precipitant. So the precipitant is a certain chemical which will help force the protein out of solution, and in an ideal world it will then become solid and form a crystal. So what happens is we mix these together and seal them above a well of even more precipitate, or precipitant even, and then the extra water will diffuse away from the drop and into the precipitant, yeah. And as that's happening, because the water concentration is going down, this is effectively increasing both our protein concentration and our precipitant concentration. And what we hope will happen is we'll start around here, and as these two both increase, we'll enter this zone called the spontaneous nucleation zone. What this means is a small bit of protein has fallen out of solution and started the very smallest semblance of a crystal. So once that happens, the protein is falling out of solution, and this means the protein concentration is then going to go back down, while the precipitant concentration will stay roughly the same. And as the more and more protein leaves solution and becomes a crystal, we eventually pass through this growth phase, and we'll end up back sort of where we started, but instead with a crystal that is a protein that is crystallized instead of in solution. And here is an actual example of one that I made. So when I mixed these two solutions together, it actually went straight into this red zone. This red zone is amorphous precipitate. So I did this, I checked my protein drop after 8 days and despaired because my experiment had failed. I hadn't made a crystal. But then after 9 days, somehow magically, it managed to fall out of the precipitation zone into the spontaneous nucleation zone. And it made this single long crystal. So I saw this on day 9. I was like, brilliant, it's actually worked. But then I didn't have any time to actually collect any data off this. And after 11 days, it had actually grown even bigger. And then 14 days, it was even bigger again. 17 days, 21 days, and then it finally stopped growing. So this is the point where we've reached this blue zone. This is called the metastable zone. So that's how we get our protein crystals. Protein crystals can be very beautiful. They come in all shapes and sizes and colors. So these crystals here are actually 2 different crystals that are kind of laying on top of each other. And then this is why you can see this nice like Battenberg-esque sort of appearance from them. And like I said, this is a green protein crystal. This contains a copper molecule. This one's red, which has got iron in it. This one here is actually insulin from a pig. So yeah, lots of, and this is probably the prettiest but most useless crystal I've ever grown, because you want a nice single crystal as opposed to this kind of 7 crystal nightmare, which I had to poke with a needle under a microscope to release the individual crystals. So we've grown our bacteria, we've purified our protein, we've managed to crystallize it. So now it's time to go to the particle accelerator. So this here is Diamond. This is where I am currently, actually. I am somewhere around here in zone 3 in my office. So although this looks to be circular, the actual machine itself is made up of a ring of magnets, and instead of being a perfect circle, it's actually a series of straights. So what it really looks like inside is more like this. So we start in the middle, which is this bit here. Oh, gone the wrong way. Yeah, this bit here. So this is the Electron Injector. So what this does is it just fires a bunch of electrons down a linear accelerator. So this just speeds up those electrons to near the speed of light. And then they move into the booster ring, where they're accelerated even more to even closer to the speed of light, before being released into the storage ring. So when electrons come in, they meet a radio frequency generator, or an RF cavity. So this is kind of akin to when you're playing Mario Kart and you hit one of those like arrow speed boosters. This just kind of catapults our electrons even faster around the ring, as they will slow down as they keep circulating. And then what happens is it hits one of these, the bunch of electrons hit one of these bending magnets. So the magnets do exactly what they say. They bend our electron beam. But what happens when we force our electrons to move direction is they actually give off radiation. So this is how we generate our X-rays. So electrons going in a straight line, they're suddenly bent, but the radiation carries on the straight line that the electrons were going in the first place. And then the rest of the ring is kind of these straight sections. So these have various magnets which kind of push and squeeze the beam to keep it straight. Then we have more bending magnets, so you can have multiple experiments going on in the different beam lines. We also have these insertion devices, which have great names. So one is called a wiggler and one is called an undulator. And they, much like the bending magnets, do exactly what they say. They make the electrons wiggle and undulate, and that actually increases the energy, or increases the, I think it's the brilliance, or the intensity of the X-rays that come off, so we can have even stronger radiation for our experiments. So once the X-rays have been generated, they are sent down another series of mirrors and magnets and slits, which kind of bend and shape the beam and eventually deliver it to our protein crystal, which will be sitting somewhere around here. So our protein crystals have been treated very badly. They've been picked up in a small loop and frozen in, I should say cooled is the proper term, because they don't change state. They're still solid. So they're cooled in liquid nitrogen, and then this robot can come and pick up our sample and plonk it on the beamline, where we can then fire X-rays at it. So this brings me to the next step, which is firing X-rays at our protein crystals. So why have we gone through all of this bother with X-rays to get a protein structure? So it comes down to sort of the fundamental physics of the electromagnetic spectrum. So we see in the visible spectrum, so the smallest thing we can see is based on the wavelength of the visible spectrum. So we can see bacteria with our naked eye with a microscope, but we can't see things that are smaller than that. So we can't see a virus particle or a protein or a water molecule. So we need X-rays, which are the right wavelength to be able to see our proteins. But the problem with X-rays is, unlike visible light, we can't see them. And X-rays also can't be bent very easily with lenses. So this is where crystallography comes in. So we can almost directly compare our crystallography experiment to your standard light microscope. So we have our light source. In visible light, it's just a light bulb. Whereas in crystallography, we have our X-ray beam. This is, if you think about this bit as all the mirrors and stuff that have got our X-rays to our crystal sample. So here is our specimen in our light microscope and our crystal in our crystallography experiment. And then as the X-ray hits the crystal, it produces this spot pattern. So this is akin to this sort of middle bit here, where the light is now being focused back into your eye. So we have to use some complicated maths to convert this spot pattern into this electron density. And then into this electron density, we build our protein model. So why do we need to make a crystal for this? This seems like it's adding an even more complicated step. Why can't we just fire an X-ray at our protein and see what it looks like? So it's a problem of signal. So I've actually used our bed sheets as an example for this. So if we've got one protein or two foxes, and we fire an X-ray at it, what we're going to get is kind of a bit of a smudge. So this is a Fourier Transform, which is just a sort of a mathematical process to change this into a pattern. And as you can see, when we've only got one fox, we just get a smudge, just like if we only had one protein, we get a smudge. If you have slightly more foxes or proteins, then fire an X-ray at them, you start seeing that we're starting to head towards actual discrete spots. So this actually holds a lot more information than our smudge. Then if we increase it even further, get even more foxes, then you can actually see now we've actually got diffraction. We've got discrete spots which hold even more information about what our protein actually looks like. And then this is an example of diffraction from an actual protein crystal. Obviously, it's a lot more complex than my foxes. So yeah, so once we've got our spots, we can use our complicated maths, which I'm not going to explain here because I only understand some of it anyway. We can then end up back at our protein structure. So proteins are kind of like the molecular machines of your body. They perform almost every process that keeps you alive. So we can think of a protein structure kind of like looking at a machine. So we can look at the entire machine and see all of it sort of functioning at once. So this is an animation of the ribosome, which is what your cells use to make more proteins. And you can see it's got lots of moving parts which end up producing your protein out the back. But then if we have a high enough detailed structure, we can look at sort of an individual component of our machine. So here is just two proteins from that large complex of proteins. And you can see here how it's bound the RNA. So we can sort of see the minutiae of how it's working. And then if we can go even further in, we can see sort of how each atom is interacting and really pick apart the kind of chemistry of how our proteins work. And one way that this has been used to great effect is in drug design. So if you have your protein structure, so say this is a protein that's gone wrong in a disease, so some mutation has happened and our protein is either working too hard or not working enough, and we need to design a drug specifically to stop this protein functioning or to make it function again. So we can take our protein structure. Hopefully this video will work, maybe not. Okay, so this is our protein structure. It's got these helices and these sheets which make up our protein structure. So proteins don't actually really look like this. They actually look more like a large blob. Come back to the start. This will come back to the start. So yeah, so this is our protein structure. I've colored it in red and blue for the charges. So we've got positive and negative. And if this will actually play now, no, of course it won't. Yeah, what happens is this protein actually is more of a blob than these actual cartoons. This cartoon is just how we represent our protein so that we as humans can better understand it. But if this animation would play, there is a kind of pocket here, which has like it's the active site of the protein. It's where it does all of its actual work. And from this structure, we were able to see a sort of a pocket with certain atoms, and looking at the natural mechanism through the structure by which this protein works, we are able to design a molecule that will fit in that space, but will actually render the protein useless and stop it working. So this protein is actually a common one that goes wrong in cancer, which leads your cells to overexpress by making it overactive. So the drug actually can sit in that pocket, shut it down, and stop your cells from proliferating. So yeah, so this is pretty much the full work through of crystallography. So bacteria to our individual protein, then we'll crystallize it, take it to the accelerator, fire some X-rays at them, and then hopefully determine our protein structure and be able to figure out how our proteins work. It is most certainly not this. So if you walk away from this talk with one thing, it's that X-ray crystallography is not artfully arranging crystals on a person's head. So yeah, with that, I will finish my talk. And if you have any questions, please feel free to ask. There's also various links here, which you can, if you want to learn more about crystallography, there's lots of resources online for how we figure out what proteins look like. You can also download the Fourier filter camera, which I use to transform our bed sheets into diffraction images. So if you have an Android phone, you can do that yourself. So yeah, thank you for listening.
Thank you so much, Sam. It was a brilliant talk. Thank you so much for the clearest explanation of X-rays that I've heard to the date, during my 4 years of PhD included. Now, if anyone would like to ask questions, please put them in the chat and I'll read them out to Sam. We have a question from Stuart, and he says, thanks for the talk. And Stuart's asking, will predictive protein folding software ever become accurate or quick enough to replace traditional X-ray crystallography and accelerate drug design?
Are you trying to put me out of a job, Stuart? So yeah, actually the Cas9 sort of competition for predicting protein folding actually it was in the news actually quite recently that crystallography is dead and we can just predict our proteins from our sequences now, which they have come a long way. Like it's amazing what they can predict. But the problem is most of their predictions are obviously based on work we've already done. So if they come across a completely new fold, they might be able to try and predict it, but then I think the problem is the predictions are exactly that, they're predictions. So if you really want a drug company to pump millions and millions of pounds into producing a drug, they're probably going to want some data behind that structure. So I definitely don't think it's going to put me out of a job just yet, but it is amazing the progress they've made. But I think the thing that structural biology probably needs to do is start actually looking more in the sort of protein dynamics range. So sort of seeing how reactions work, like really doing the kind of nitty-gritty chemistry side of it, which you wouldn't really be able to do with a predicted folded structure.
Brilliant, thank you so much, Sam. I had a question actually. So you said that your degree was in medical biochemistry.
Mm-hmm.
So how did you find the sort of transition from sort of the biomedical science really to sort of very heavy biophysics and maths-based science?
Yeah, I mean, I guess the good thing about working in crystallography is it kind of spans a lot of fields. So when I started my PhD, I was still doing biochemistry, like I was doing all the protein side of things, and then I learned the kind of, if I'm honest, I didn't really properly learn the X-ray physics until I was writing my thesis, and I had to write a chapter about it. So yeah, you kind of start with what you know, and then kind of build on it from there. Me and my partner actually joke about this. She has more of a formal physics training than me. She did A-level physics, and I didn't do it at all, and yet I ended up somehow working in physics, despite having no training. And I did history, and she ended up working in history. So it's weird the places you can end up from a biochemistry degree. Definitely,
and I think just in general, we as scientists, we now see how interlinked everything is. So you're never going to be just this one discrete person doing one thing for the rest of your life. You'll be changing, and I think it's very important to communicate.
Oh yeah, I mean, Diamond would absolutely fall apart if I was expected to do all of the physics, all of the engineering here. Like it's one of the great things about working here. Like you get to have a meeting with the engineer and say, I've got this idea, and then they'll actually be able to make it a reality from your stupid idea.
That's brilliant. Do we have any more questions from the attendees? Okay. Rosemary, would you like to bring the session to the close then? Oh, we have actually a question from Christine, if that's okay. Christine is asking, why do helical structures produce a cross pattern of dots? Thinking of DNA, but do these apply to alpha helices?
So I think the thing about DNA is it's really obvious to see the kind of cross pattern, because the structure of DNA is just that. It's just the helix. So when you've crystallized your DNA, you're going to have lots and lots of those DNA molecules repeating. And because it's such a simple molecule, really, you actually can see the kind of cross pattern in the diffraction. So if you look up Rosalind Franklin photograph 51, you'll be able to see the cross pattern that we're talking about. But in a protein structure, because you've got so many helices going sort of in crisscross directions, and then you've got sheets as well, you don't get this kind of nice defined single feature, because everything's kind of twisted together.
Thank you, Sam. Yes, proteins are just very complicated. The directionality and the amorphous structures. Okay.
Yeah, so if you did have a protein that was sort of really small and just helical, you might see a similar sort of diffraction pattern.
That's brilliant. Okay, thank you very much, Sam, and I'll hand over to Rosemary now.
Right. Thank you very much, Sam. Very, very interesting. Proteins really are everything, aren't they? But the interdisciplinary work, lots of different scientists working together, make for great science, basically, I think. So thank you, all of you in the audience. And some of you I know this is your third time, and I do hope to see you and everyone else tomorrow, please. If you can make it, it would be lovely. Dr. Gareth Starbuck is coming from, not coming from Nottingham, he can appear on our screen tomorrow, Nottingham Trent University, and he's going to give a talk, Genetics, Sex Selection and Conservation. So something else to look forward to, and we all look forward to seeing you all again tomorrow. Thanks very much for coming, and thank you, Sam, again, very much. Thank you. Bye-bye.
No problem. Bye.
Bye.